Hello everyone, this is Dr. Meghla. Welcome to my lecture in glycolysis. The topics to cover under glycolysis were introduction to glycolysis, steps involved in the pathway, regulation of pathway, energetics of pathway, inhibitors of pathway, and fate of pathway. Glycolysis is said to be EMP or Embden-Meyerhoff-Parnas pathway. This pathway is found to occur in all the cell cytoplasm. The pathway is said to be a unique pathway of carbohydrate metabolism where glucose is oxidized to yield pyruvate or lactate. The pathway is said to be unique because it can carry out in the presence of oxygen as well as in the absence of oxygen. So aerobic end product will be pyruvate and anaerobic end product will be lactate. Glycolytic pathway involves 10 steps. Out of these 10 steps, reaction 1 to 5 we call it as preparative phase, where 2 molecules of ATP were utilized to synthesize 2 molecules of 3-carbon glyceraldehyde 3-phosphate. Reaction 6 to 10, we call them as payoff phase, where ATP was yielded and 2 molecules of 3-carbon glyceraldehyde 3-phosphate is converted to 2 molecules of pyruvate, which is the 3-carbon compound. The net yield of ATP per molecule of glucose metabolized in this pathway is found to be 8 if it is an aerobic respiration, and in case of anaerobic respiration, the yield of ATP is found to be 2 molecules.
We now look into the steps involved in glycolysis. Glycolytic pathway involves 10 steps. Step 1 to step 5 is said to be the preparative phase, where one molecule of glucose on oxidation will be yielding one molecule of a 3-carbon compound called glyceraldehyde 3-phosphate and one molecule of 3-carbon compound dihydroxyacetone phosphate. So if you check step 1 to step 5, in step 1 and step 3, there is one molecule of ATP is consumed for the reaction. To begin with the glycolytic pathway, glucose is converted to glucose 6-phosphate by hexokinase, and this is said to be the rate limiting step, and this reaction is highly irreversible. Glucose 6-phosphate is then converted to fructose 6-phosphate, catalyzed by an enzyme phosphohexose isomerase, which is a reversible reaction. Fructose 6-phosphate is converted to fructose 1,6-bisphosphate, catalyzed by an enzyme phosphofructokinase 1. This is said to be a second rate limiting step, and this reaction is also highly irreversible. So far we have seen with two irreversible reactions. Fructose 1,6-bisphosphate is converted to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, catalyzed by an enzyme called aldolase. So in the next step, where dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate, catalyzed by an enzyme called triose phosphate isomerase. So we have yielded with 2 molecules of glyceraldehyde 3-phosphate. Now, glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate, and this reaction is catalyzed by an enzyme called glyceraldehyde 3-phosphate dehydrogenase. Here, there is an involvement of 2 NAD coenzyme for this reaction, and thereby there will be production of 6 molecule of ATP in this step via oxidative phosphorylation. Now, the product formed called 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate This step is catalyzed by the reaction enzyme called phosphoglycerate kinase. And here an ATP is formed at the substrate level itself, and hence we call them as substrate level phosphorylation. In the next step, the 3-phosphoglycerate is converted to 2-phosphoglycerate catalyzed by an enzyme called phosphoglycerate mutase, and this is a reversible reaction. 2-phosphoglycerate, then it will be dehydrated with the release of 2 water molecule converted to phosphoenol pyruvate catalyzed by an enzyme called enolase. The phosphoenol pyruvate which is formed is converted to 2 molecules of pyruvate catalyzed by an enzyme called pyruvate kinase, and this reaction is irreversible, and you could see 2 molecules of ATP generated at the substrate level, and hence we call them as substrate level phosphorylation. Here comes the overall reaction of glycolysis. So this is for aerobic oxidation, where glucose on oxidation yields 2 molecules of pyruvate with the involvement of coenzyme NAD. And in anaerobic oxidation, glucose will be yielding 2 molecules of lactate. Coming to regulation of glycolysis, we have seen with 3 irreversible reactions in glycolytic pathway, and these 3 irreversible reactions were playing a major role in regulating the glycolytic mechanism. Coming to regulation of glycolysis in muscle, due to the availability of high energy compound ATP, the regulation of glycolysis in muscle will vary. At rest, there will be high energy ATP will be available, and thereby the ATP will be inhibiting an enzyme called phosphofructokinase, and they are inhibiting an enzyme called pyruvate kinase. Similarly, glucose 6-phosphate is inhibiting hexose kinase. In case of severe exercise or strenuous exercise, the availability of high energy compound ATP will be low, and thereby it is favoring the activity of an enzyme called phosphofructokinase and pyruvate kinase. Coming to the energetics of glycolysis, the energetics can be made in the form of a table where the pathway is glycolysis, the reaction catalyzed by, under which you can write the reactions catalyzed by an enzyme that is responsible for either substrate level phosphorylation or they are involved in respiratory chain or electron transport chain. In case of the enzyme glyceraldehyde 3-phosphate dehydrogenase, it results with the production of 6 molecules of ATP. Similarly, the reaction catalyzed by phosphoglycerate kinase, where you yield with 2 molecules of ATP at the substrate level. Similarly, a reaction catalyzed by pyruvate kinase ends with 2 molecules of ATP at the substrate level itself. Therefore, the net yield will be 10 molecules of ATP in aerobic oxidation per molecule of glucose oxide. But please do remember that we have been utilized 2 molecules of ATP in steps 1 to 5. So you need to subtract 2 molecules of ATP that was consumed in the beginning of the reaction, that is in step 1 and 3. So if you subtract -2 from 10, the net yield of ATP will be 8 molecules. That was the yield of ATP molecule per molecule of glucose oxidation under aerobic oxidation. So coming to inhibitors of glycolysis, arsenate and iodoacetate were said to be the inhibitors of the enzyme called glyceraldehyde 3-phosphate dehydrogenase. Bromohydroxyacetone phosphate is an inhibitor of dihydroxyacetone phosphate. Fluoride is an inhibitor of an enzyme called enolase. Oxamate is an inhibitor of an enzyme called lactate dehydrogenase. So this is the enzyme catalyzing the anaerobic glycolysis. So talking about the fate of pyruvate, we have three different fate for pyruvate. Pyruvate has been converted to lactate during anaerobic glycolysis. You required an enzyme called lactate dehydrogenase. So pyruvate is converted to lactate with the help of lactate dehydrogenase enzyme, and you required NADH2 for converting to lactate. So the other fate of pyruvate is conversion of pyruvate to alanine. Alanine is an amino acid. So pyruvate is converted to alanine with the help of an enzyme called alanine aminotransferase, which takes up glutamate and converts alpha ketoglutarate and alanine. So the other fate of pyruvate is that pyruvate is entering into the TCA cycle with the help of pyruvate dehydrogenase complex, where pyruvate is converted to acetyl-CoA and it requires coenzyme A and it requires coenzyme NAD. Thank you.